Thank you so much for coming, and thank you again to the organizers of this conference for having us here. It's been fantastic so far. And I want to echo the words that were said about Brain and Behavior Research Foundation. On the outside, people that aren't directly involved, this has an impeccable reputation as being one of the premier places to go. To get an award from here is a huge feather in the cap, as well as something that can be used to push and really work on science that is a bit out of the box, the kind of stuff that the government probably would not fund or not fund easily. And it's really, on multiple occasions past, have made really good things happen. So I want to echo that. So my talk today is about genetics. We're going to be talking about where we are in schizophrenia genetics and where we need to be. I'm going to tell two stories today. The first one is about breaking the cycle. So as I think everyone here probably knows, but let me go over it real quickly, the impact of schizophrenia in any way you want to look at it is massive. The impact upon morbidity, in other words, the non-death-related causes of illness, places it in the top 10 in the world. In terms of mortality, people with schizophrenia tend to have a much shorter lifespan than others. And the cost in every way you want to look at it, personal, familial, societal, monetary, is enormous. And the one part of that we can actually put a number on is the monetary cost, which has been estimated to be on the order of $1.4 million per person per life. And we've done a lot of things to go after schizophrenia. I mean, every way of looking at it that one can think of has been applied. Some of these things have not worked out in the long term. Others actually provided really important and really interesting hints. But I think that really what's going on is very, very likely a very subtle process rather than something that's really dramatic and overt. And I think over the years, genetics has emerged as a really, really important clue. But one of the key things, again, is that it's a subtle process. So what's going on is certainly a probabilistic process rather than something that's deterministic. To the best of our knowledge, there is no gene for schizophrenia. Rather, one inherits a predisposition, and that predisposition may or may not be manifest over time, over life. So for example, the risk of schizophrenia is on the order of 1%. For people with a positive family history of schizophrenia, a first-degree relative with schizophrenia, their risk might rise to 10%. So you go from 1% to 10%, which is a very large relative risk, but the absolute risk is not huge. And I think one of the best figures of this that I know of is if you have one identical twin with schizophrenia, the risk to the co-twin is on the order of 1/2. And again, I think this comes together to indicate a subtle process. Now, I think if we go back and be honest about it, I think schizophrenia genetics was a bit stuck. We did not know what was happening in terms of the architecture, in terms of the number of different genes, how big their effects were, which means that we could not actually design the proper study. There's an unknown impact of diagnostic heterogeneity. In other words, if we look at 100 people with schizophrenia, are there one type of schizophrenia, or 10 types, or 20 types, or even 100 types? Not knowing that also was a problem. When we actually looked at the genotype, at the DNA of individuals using genotyping and understanding where the risk might be, the technology was limiting. We weren't able to do a lot, just a very few places. And our samples historically over time have been actually quite small, and that meant that power was low. We did not really have the resolution that we needed. So in 2007, a number of us founded the Psychiatric Genomics Consortium, the PGC. I guess I'm the lead PI of the PGC, and really my job description is herding cats. That's what I do professionally more than anything else. And one of the reasons we did this was because the sample sizes that we were going to need, we anticipated were going to be large, far larger than any of us individually had. And that meant that cooperation was the only way forward. When we set up the PGC, it was from the very start meant to be a democratic, open, participatory, collaborative thing, which really helped us get going. But rapid progress was really a major, major impetus for us. This is the structure of the PGC right now. I chair the coordinating committee. We have 9 disease groups: ADHD, anorexia, autism, bipolar disorder, major depression, OCD, Tourette's, post-traumatic stress disorder, schizophrenia, of course, and drugs and alcohol. Schizophrenia is chaired by Mick O'Donovan, who was an awardee of the Lieber Prize, I think, 2 years ago. We also have additional groups that sort of do things which pertain to all the different groups. So in terms of some highlights of what we've done so far, first, we are the largest consortium, to my knowledge, in the history of psychiatry. Currently, there's 800 people or more in the PGC. It's easily the largest biological experiment in psychiatry. If the information we get at the end of it is 1 pixel on a screen, the amount of information that goes into it would be 772 miles wide on that scale. We've come up with multiple high-profile papers over the time. Currently, there's 172,000 individuals whose genomes are actually in analysis right now. So 172K. We're currently doing genotyping to increase that number by 100,000. And we're hopeful, and our goal for the PGC is we want to get 100,000 cases for each of the 9 disorders. That's our next crazy goal that we're going to try and shoot for. And that will give us, in the end, somewhere in the order of 1 million people that we're actually analyzing. So the first story is a new model of science. What we've tried to do in the PGC was to create something which, there certainly had been examples before, but never in psychiatry, where we're very cooperative, collaborative, comprehensive. Innovation is really, really important, because to do a lot of this stuff requires us to think out of the box and find new ways to get to solutions. I think the reason it's worked is because most of us are clinicians, and we realize that we can't get there unless we work together. So there's a really important degree of altruism involved. And obviously, it's open and democratic. And the cool thing about this, in many instances, is this incredible can-do attitude of people in the consortium. The second story is looking behind the curtain, the Wizard of Oz metaphor. So if we actually, what have we learned? What have we found out about schizophrenia in particular? So I would argue that for many people, they think they've got a handle on what's going on in genomics and schizophrenia. And I would argue that an update's needed. Just a bit of a caution that this is a really, really active area right now, which is fantastic. A lot of what we know actually isn't published yet, and it's in the process. And at the same time, even as we're writing papers to tell what we know right now, we're doing a bigger study behind it that will come to fruition in a year or so. So I would add a caution. So let's just of course, be skeptical, but do keep an open mind about where we are. So how can we use genetic data? And there's 3 uses. I'm only going to talk about the first one, about etiology, the causes of schizophrenia, that it can point at biology and can give us some idea as to where our pharmacological colleagues could actually intentionally intervene. We could also talk about therapeutics to predict adverse drug reaction to treatment response, and also how we can use genomics to stratify samples. I'm not going to cover those in any detail. If people are interested, please ask me during the Q&A session. So what's the logic? What are we doing this for? And I think I'd start off by saying that drug discovery in psychiatry has mostly been serendipitous. There have been just sort of happy accidents that have led us to the major drug classes that we use. A few exceptions, but generally that's the rule. The idea that we have is to use genetic findings, hardcore, rigorous, robust, replicable genetic findings, to give us an idea about the biology. Now, in some instances, the biological implication will be obvious. In other instances, it will require additional work, hence the little red dagger there. But that will give us then the pathways or the biological readouts, what is going awry in neurons or during development in some way that eventually gives rise to schizophrenia. If we know that, then that's where we can actually begin to do rational drug discovery, which really is the goal of the whole exercise. So there's something called exome sequencing, and this is sort of, this technology is such that if I have told you what we could do 10 years ago, you would have thought it's science fiction. Right now it's routine. It can be applied in most major hospitals in the world. And essentially what it entails is taking all the parts of the genome that actually make proteins and going back and looking at every single base over and over again until we get a good idea as to what's there. We can do that in one individual. The cost these days is in the order of $1,000. There's three small studies, 2 large studies. These guys were in Nature about 6 months ago. And the net of it is that no single genes emerged as coming up as being involved in schizophrenia. However, a bunch of pathways came up. Calcium signaling, the postsynaptic density activity-related cytoskeleton complex, a really key part of neuronal function, NMDA receptors and genes whose products are bound by something called the Fragile X mental retardation protein. These things came up in a whopping way. To my knowledge, at present, there's no single gene Mendelian disorder. So we've yet to find a single gene that really carries super high risk for schizophrenia. There's a bunch of looking. Perhaps that'll come up, but it's, to my knowledge, has yet been identified. So even this is what's published right now. In analysis right now are much, much larger samples. Now, as it turns out, usually we have 2 copies of DNA, one from mom, one from dad. There are instances where you might only get one, and this is called a copy number variant or a copy number variation. And 10 of these have been identified, which are really quite rare but quite potent. And in fact, I think it's to the point that if you were to actually screen people, who came in through the door with a clinical diagnosis of schizophrenia in a hospital setting, you might actually find that 1% to 5% of patients would actually have these. Some of these things it's important to know about because there might be some physical comorbidity that goes along with it, and a lot of us have wondered about whether this would be a very useful routine thing for us to do. And there's a whole bunch of new cases in analysis, and it may be, in fact, that all the very large events are the ones that are listed here. Now, the next technology we've tried, the next approach is GWAS, Genome-Wide Association Study. Here, we're looking for common variants, things that are present in most of us, but have a subtle effect individually. But as it turns out, there may be hundreds, perhaps even thousands of these. And there was a paper we published in the journal Nature about 6 months ago, and given the nature of the consortium, there were 302 authors. One of our colleagues actually had to type them all into a website one at a time. And about halfway through, he sent me a message that only said death. So Stefan will be talking about this, I think, 2 talks from now, but this combined analysis of almost 37,000 individuals with schizophrenia, 113,000 controls, and in the end, we discovered 128 different associations and 108 strictly conservatively defined genomic regions. This was an increase by a factor of 5 from the previous paper that Stefan and I and others had published about a year before. And he'll be talking about this, but this is something called the Manhattan plot. You have the genome from chromosome 1 all the way up to chromosome X, and how high something is how significant it is. And this bar is the significance line. And there are 108 different regions over here that actually met the grade, that actually made a really rigorous idea of significance. This is meant to look like Manhattan from across the East River. And we've certainly seen these before where it really looked like, Omaha or something, nothing. So this is actually really cool. And Stefan will talk about this. But if we look at the specific associations, DRD2 popped up. This is the dopamine 2 receptor, which is the target of pretty much all antipsychotics. So it's a real great idea that this has some validity, or this might actually point to something that's actionable in terms of drug development. NMDA receptors popped up, calcium signaling. There's a really, really interesting story that this is Steve McCarroll's to say, so I won't mention it here, but this is the hardest part of this, the hardest association we have, the tallest one in the Manhattan plot. Steve McCarroll and his colleagues appear to have figured out. It's a spectacular story with a lot of interesting nuances, and that's hot off the press. But the cool thing is a lot of these things converge. So when we look at the protein coding parts using exome sequencing, copy number variation using different technology, common variation using GWAS, the same things tend to pop up. Neuronal calcium signaling and a couple of different compartments or neuronal processes pop up, which is really interesting because it's difficult to understand that in the form of bias. Most of us think this indicates that there's a really strong whiff of truth here, which obviously is what we're trying to find. Now, we've designed something called the site chip. This is led by Pamela Sklar, who's at Mount Sinai here in New York, and Ben Neal at the Broad. And this has 550,000 genetic markers that 50,000 of which we've selected ourselves. And basically, this is cheap. It's $100 to do it. In my lab, we actually use it for quality control. We've made it so that anybody can purchase it in the world and use it for their own purposes. But currently, the PGC is doing 100,000 new samples, and we're hopeful that we will have 62,000 samples with these data for people with schizophrenia next year. So the previous study, the Nature paper from July, had 36,000, 37,000 cases. We're hopeful to have at least 62,000 shortly. So the second story is what we've learned. Genomics works for schizophrenia, and that's critical. And the more we do, the more we actually push this, the more we learn. So I think we finally found an approach that if we stick to it and push it hard, it'll actually reveal a lot of the important stuff that we need. So coming back to my title, where are we in schizophrenia genetics? So I would argue that genomics is really starting to deliver spectacular new insights. I wish I could say more about the Steve McCarroll stuff, because that's actually the coolest one here. So we'll have to keep an eye for that. But it's very, very interesting. But we're starting to learn what schizophrenia really is from the perspective of the genome, how nature wrote this disorder, what it is, what it isn't. One of our colleagues said that the Nature paper was sort of a nice pilot study. And this is certainly something that he said it with tongue in cheek. We learned a lot, but really it is just a step rather than an end. And if people are interested in N01 studies, for the interest of time, ask me at the Q&A, and I can come back to that. But where do we need to be? We need to have a far more complete understanding of schizophrenia. We need to really, really crack it open and really understand with as much nuance as we can what it is. And we need to find the pathways, we need to get the readouts, and then obviously move to compound screening. Now, the interesting thing is, I would argue that this could be done, the genetic architecture of schizophrenia could be determined on the 3 to 10-year horizon. This is actually kind of an interesting statement, and I believe it's something we can actually back empirically. This is not just me ranting up here because I want money or something. It's unprecedented that we can say that, but I believe we can prove it. All we need to do is apply existing technology that actually has become extremely affordable. There's no magic that's required, no technological stuff. It's essentially pushing what we've done so far in a logical, rational, sensible way. And we just need the resources to do it. In fact, one could argue that if this work were eventually to prevent merely 30 cases, to prevent 30 individuals from having schizophrenia, it would be worth it from a financial perspective alone. Now, the other sort of thing which I think comes from this is hope. I know a lot of people in the room have family members or close ones, or maybe even themselves have been in fact impacted by this illness. We're working really hard on this. This is really, we've got a lot of top-flight people that are involved in this. It's something that we're pushing as hard as we can and as quickly as we can. So I believe there's some interesting hope here too. The other thing that I can't go into in great detail, but there's also this there but for the grace of, fill in the deity that you prefer, go I. As it turns out, most of us inherit at least some schizophrenia risk alleles, some genetic variants that inherit it. Some people in this room who don't have schizophrenia have actually inherited a lot of them. And I think this is something which will actually place this illness in a different way. There's a real interesting opportunity for destigmatization here, the knowledge that we actually all have at least some propensity to this illness. So finally, I'd like to thank BBRF, the Liebers. NARCED had kindly given me a Senior Investigator Award some years ago, which was extremely helpful in doing some cool stuff. Obviously, I've got, my lab people are fantastic, and they're as much a part of this work than I am. My colleagues in the PGC, particularly Mick O'Donovan, Stefan, Ben Neal, and Mark Daly have been absolutely fantastic, as well as the other 795 individuals. The Swedish Research Council has been very generous in the funding. The PGC has been funded by many different sources, including BBRF. But of course, I also want to thank my family. This is standing with glass and 700 feet straight down. That's my wife, Cynthia Bulick, who's a renowned academic in her own life, daughter Emily, son Brendan, who actually is working in genomics right now, daughter Natalie. And this is the guy who is faking a jump because he's actually scared out of his mind. And then finally, I come from a fairly nerdy family. This is, when you do a study, you have to get permission. You have to demonstrate you have training in IRB stuff. And I did this, and actually all my family's on here, so we're a family of nerds. Thank you so much for your attention.